Good morning, everyone. Thank you for coming on time. We will get started right away. We have a lot to cover for this session. So last week we learned about the scientific methods, and we talked about how to set up an experiment, how to look at different sources to see whether they're reliable or not. And today we're going to shift gear. We're going to actually talk about actual biology now. And you might remember last week we talked about the organization of life, right? Where we started with atoms and molecules, and then they go up to the cell level, tissue, so on and so forth. So as we progress through the course, we're going to go from small things to eventually the entire organism. So now we will start by talking about macromolecules. Okay. So what are macromolecules? Well, there are four major macromolecules in the in living organisms. Okay, so we have the carbohydrates, which are primarily used as energy source. We have lipids that makes up our cell membrane, for example. We have proteins, and that has various functions, including structure, communication, to say a few. And we also have our nucleic acids. So these are macromolecules. These are really big molecules that have thousands and thousands of atoms in them, compared to something like water, which is, as you know, H2O, that only has 3 atoms. So macromolecules have thousands of atoms in them. Okay, so let's take a look at the first one here. List 5 major elements found in living things. Okay, so we have things like carbon. Okay, carbon. Let me choose a different color here. We have carbon. Okay, and the symbol for carbon, as you might know from chemistry, is C. Okay, we also have a lot of hydrogen. That is H. Nitrogen, phosphorus, and of course we have oxygen as well. So these are the 5 major elements that makes up an organism. Okay, we also have what's called trace elements. Does anybody know or remember from the lecture recordings what trace elements are? Anybody? We only have 4 people here, so don't be shy. 5 people now. So what are trace elements? No one knows what trace elements are? Okay, so trace elements are elements that exist in very small quantity in the body. Okay, so if you have too much of it, is going to be usually going to cause some problem, and if you have not enough of it, that's also going to cause a problem. It's one of those things that you need just a tiny bit in order for the body to function at the right level. So example include things like zinc. So zinc is Zn. This helps you with your immune defense. We have calcium. Right? So calcium, you need that for bones, for example. Magnesium. It's an important electrolyte. And finally, we have maybe iodine or something. Okay, there are many others, right? Iron, right? That's another example. Right? So iodine, you need it for your thyroid gland, right? To make the metabolic hormones, right? They're called T3 and T4. Yes, and iron. Okay, I'll put iron here as well, since Nadia wrote it down in the chat. We need iron to make hemoglobin, for example. And that's something that helps us carry oxygen in our red cells. Okay. All right, so this feels more like chemistry first, but then you got to understand the chemistry before you understand the bio molecules. So this will feel like it's a chemistry class for a little bit. So when atoms come together and form a stable particle, we call that a compound. Okay, compound. So put atoms together, you get a compound. Now there are two types of compounds that you can form. If you have a metal plus a non-metal, then you will get an ionic compound. So if you look at the periodic table, and I don't have one for display right now, but on the periodic table, there's like a little staircase here. So typically, generally speaking, the elements that are on the left of the staircase, those are the metals. Okay, and then over here, these are the non-metals. So if you take something on the left side, so to speak, and you combine with something on the right side, then you are going to form an ionic compound. Okay, so these kind of bonds, a bond is what holds the atom together, right? So that's going to be an ionic bond, ionic bond. Okay, so again, right, for those of you who are not sure, bond is just what holds the atoms together. Okay, so ionic bond. Now, this kind of ionic bond, right, involves the transfer of electrons from metal to non-metal. Okay, so metals, they don't really like electrons. They don't want to give them away. For the reason you can watch the recording. Remember, this is just a review, right? Review of what you saw in the video. And the non-metals, they love electrons, okay? So when a metal meets a non-metal, it's perfect because the metal will give the electron to the non-metal, and in the process, they will form an ionic bond. Okay, so these ionic bonds are really, really strong, and that makes ionic compound solid at room temperature. Okay, so example of ionic compound would be things like NaCl. Right? NaCl is sodium chloride, and that is table salt. Table salt. Okay, another example of ionic compound could be like sodium fluoride. Okay, so this is the stuff that is found in your toothpaste. And that's why your toothpaste is a little bit salty, right? And the fluoride helps protect the teeth. Okay, so you can create any combination as long as it's metal with a non-metal, right? Calcium chloride, right? A bunch of other examples. On the other hand, molecular compounds, it's something that you will form when you take a non-metal and you combine it with other non-metals. So non-metals only. Okay, and in this case, instead of forming an ionic bond, you're going to form what we call a covalent bond. Covalent bond. Okay, so what's holding the atoms together? Are these covalent bonds. And instead of transfer of electrons, they are just sharing electrons. Sharing of electrons. Okay. So unlike the ionic counterpart, the molecular compounds could be solid, could be liquid, or even gas in room temperature. Okay. So example would be water. Okay. That is H2O, you know that already. Carbon dioxide. Right, so that would be like a gas at room temperature. This would be liquid at room temperature. And then it could be like C6H12O6. Anybody know what that is? Anybody took biology before, or you remember reading about it? What is that?
Sugar?
That is sugar. Very good. So the fancy word for sugar.
Glucose.
Sort of, yeah, glucose, exactly. Okay, so these are examples of molecular compounds. All right, moving on. Indicate the polarity of water. Okay, what is polarity? Okay, polarity means there is an uneven distribution of charges causing part of the molecule to be positive and another part to be negative. So there could be like no polarity, which means it's, neutral all around. Okay, so polarity basically means does one part of the molecule or like is one part of the molecule more positive than the other? Okay, and if the answer for this is yes, then we say the molecule is polar. Okay, and then if the answer to this question is no, then we say non-polar. Okay, now do not confuse polarity with charge. Okay, if I say like, Cl minus, it means it has an extra electron. So this one is charged. Okay, like this is a charged particle. But if I just say polar, then overall this molecule is neutral. Neutral means no charge. And no charge means no extra electron or missing electron, which means, electron equals to protons. Okay, so overall it is neutral. Okay, it doesn't carry a charge. But, and this is very important, this is the question that only 20% of the people got right on the quiz last year, last semester, whatever. The molecule is neutral, but it can still be polar. Okay, because in this case, this bond here, it's a covalent bond, right? And it's covalent bond because hydrogen and oxygen are both non-metals. So what this bond is showing is that electrons are being shared, right? Okay, but the sharing is not an even sharing. Okay, think of like two siblings. Okay, like my daughters. I have two daughters. Okay, I bought them a new toy, like one of those boogie boards, a writing board, and I say, okay, you guys have to share. Only if you share, I will get it for you. Okay, so they are sharing, but then the older one, it's always hogging it. She's like always playing it more. Okay, so even though they're sharing, it's not an even sharing. It's not like half-half. So similarly, even though the electrons are being shared here, the oxygen, it's like my older daughter, it's like hogging the electron. Okay, so the electron spends more time around the oxygen than it does in the hydrogen, and as a result of that, there is a slightly negative charge. Okay, so this symbol is like sigma. Okay, it means slightly. So slightly negative on the oxygen because the electron is being pulled towards it, and then the hydrogen will be slightly positive. Okay, so overall, the molecule is polar because part of it's more negative compared to the other, but overall it is neutral. So if you have to describe a water molecule, you would say it is neutral but polar. Any question? And you're like, so what does this all mean if it's polar? Well, let's draw two water molecules right next to each other. If I have one water molecule here, okay, so we can draw like H2O if you want. Okay, usually people just draw like Mickey Mouse. Okay, so like this would be the oxygen here, and then this would be the hydrogen. Okay, and then you can draw another Mickey Mouse here, oxygen, hydrogen, like this. Okay, so what happens is When they are next to each other, one part of the water molecule, the slightly positive part, will now be attracted to the slightly negative part of another molecule. And that is going to form an interaction. Okay, so like if I have another molecule here, another water molecule here, right, it will be attracted to the oxygen, because this is slightly positive and then this is slightly negative, right? So these interactions that are between molecules, we call them hydrogen bond. Hydrogen bond. Okay. So the more hydrogen bonds you have, the higher the boiling point is going to be. Right? And as you know, water has a really high boiling point, 100. Okay. So don't confuse these hydrogen bonds with ionic bonds or covalent bonds. The ionic bonds, covalent bonds are the interaction between atoms within a molecule. The hydrogen bond, I'm telling you right now, is interaction between two molecules, okay, rather than between the atoms that is found within the molecules, okay? Use diagram to explain how ionic compounds such as NaCl are soluble in water. So you might now know, right? NaCl, right? NaCl. NaCl is actually made up of, like if you put it in water, so if you put solid and then you put it in water, it's gonna dissociate into Na plus, and then we put AQ because it's gonna be in water now, AQ like that, right? It's gonna dissociate, it's gonna dissolve, right? So that's how you use a chemical reaction equation to represent dissolving. So not everything is gonna dissolve in water, but NaCl will. And once it dissolves in water, what happens is you have the Na plus here, and then the oxygen part of the water molecule will be surrounding the Na plus, because that's the negative portion of the water molecule, right? And then you have the Cl minus here, and then over here, it's the other way around. You would have the hydrogen surrounding the chlorine. Okay. So if you think about something like, hydrocarbon, okay, something like propane, okay, or yeah, let's do propane. CH3, CH2, CH3. Okay, so you have this thing that is non-polar. Okay, there's no charge on it or no uneven distribution. And what happens if you put this in? Then the water doesn't know what to do with it. It cannot surround it like it does for the NaCl. And so something like this is going to be hydrophobic, right? So in the lecture, I told you hydrophobic means afraid of water. It's not going to mix well with water. Any questions so far? Okay, now I sent an email yesterday because I noticed like a lot of you are just joining the class, late. So can you quickly do let me know, like on the poll, like if this is your first class with me, can you say A? If you were here last time, then you can say B. Okay. Loveline, are you trying to say something? Because I see your mic moving, but I cannot hear what you're saying. Okay, so only a few of you were not here last time. Now, I'm gonna quickly show you guys something. Because I want to tell you, it is very important for you to watch the recording before coming to class. So I can actually see who has watched the recordings. Okay, and over here, these are the lecture recording for today's class. And of the 17 people in the class, only 3 people watch the first 2 parts, and then only 2 people watch the last part. I mean, if I click in, then I would know who it is, but I'm not going to do that right now. But it's very important that you watch the recording first before coming to the tutorial, because in the tutorial, I'm only giving you the condensed version, right? And I'm going through concepts quite fast. So sometimes you might not know what I'm talking about if you haven't already watched the recordings. Okay, so I urge you to do that. Now, if you're not sure where to find the recordings, let me show you one more time. You go to content and then lecture and then lecture 2, right? Next week is lecture 3. So you press on lecture 2. There are the lecture recordings. Okay, so you download the PowerPoint, you watch the recordings, and then you download the study guide before coming to the tutorial. That is very important. Okay. And since most of us are here, let's quickly talk about the quiz. We do have a quiz next Tuesday. It will be available to you from 10 to 10. So you when you're ready, you will click on this link, okay, and it will take you to the quiz. And once you're there, I mean, I wouldn't be able to show you because I'm in teacher's mode, but let me change to student mode. So next Tuesday, when you're ready, you don't have to do it at 10, okay? Like if you're busy in the morning, you can do it at like 2 p.m. or whatever. As long as you do it between 10 to 10, then you're okay. Then you click on it, and then now you cannot start because we're not within that window. But then on the day, there will be a start button, and then you click start. Once you start, the timer starts clicking, and you have to finish before the timer stops, okay? Even if you close your browser, it will keep on going. So make sure you are going to be undisturbed during the quiz period, okay? Now, there are going to be, I don't know, you can add up these numbers to see how many questions. What is this? 22 plus 5, so that's 27 questions. And you're going to have the multiple choice question, 11 multiple choice from lecture 1, 11 from lecture 2. Lecture 2 is what we're doing today. There are application question and multi-select for lecture 1, but not lecture 2. Okay, so the whole thing is out of 30. If you have an IEP, right, in college, we call the center that deals with IEP, we call that the CALS, C-A-L-S. I forgot what it stands for. But if you are registered with them and you need accommodation, please send me the documentation so you will get extra time. Okay, so this particular quiz is going to be 45 minutes. Does anybody have any question before I go back to the study guide? No? Everybody's happy? Okay. Next, okay, comparing acid and base. So what is def? Yes, Michelle, go ahead.
For the quiz, if we have any questions, would you be available for us to get in contact with you, or?
I think the questions are fairly self-explanatory. I haven't had a situation where a student is like, "Oh, this question doesn't make sense. Like it has a mistake in it or something," right? So but you can email me. I'm like usually around my phone all day, so you probably will be able to reach me. But I mean, if for some reason you find that there is a problem with a question and you think there is no answer, just try your best first, and then take a screenshot or picture of that particular question and email me after. So if there is indeed a problem with it, we can address it afterwards. Okay, but you can try emailing me. I will try to, reply you on the spot. Okay.
Okay, Maurice. Thank you.
Yeah. All right. So where are we? Definition of acid. Okay. So what is an acid? Acid is a compound that produces H plus when dissolved. Oh my goodness, not enough space, in water. Okay. Base, on the other hand, is a compound that produces OH minus when dissolved in water. Okay, so this is kind of like a simplified version of what it is. Now, pH is a scale that we use to measure the acidity of a substance. Okay, so if you have a liquid here, you can dip a pH meter in, and it would tell you, okay, whether it's an acid or base. So the pH scale goes from 0 to 14 in general. So anything that is less than 7, so pH less than 7, we call it an acid. If it's greater than 7, then it's going to be a base. And if it's at 7, then we say neutral. Okay, so distilled water is neutral. Properties, acid would be like sour. Okay, so think of like orange juice or vinegar. Those are acid. And corrosive. Base would be bitter. So bleach, or not that you should taste bleach, but something like soap, right? Like, you know, tonight, if when you're showering, you can just taste your soap a little bit. It's going to be bitter because it's basic. Most cleaning products are basic. And then corrosive, okay? Like think about bleach. You pour bleach onto like metals, it will corrode the metal. So example, we said like vinegar, okay? And then we have bleach, right? Or like ammonia. Right, so all these are example of acid and base. Okay, so that's it for the chemistry. Finally, some biology. List the 4 types of macromolecules found in the body. So I told you already, right? We have carbohydrates. We have lipids, proteins, and nucleic acid. Okay. So there are different types of carbohydrates, different types of proteins, so on and so forth, and we're gonna go through the study guides and review them. All right. Now, macromolecules, they have a common theme that they share. All right, because they're really, really big. They are actually made up of multiple repeating units. Okay, with the exception of lipids. Lipids doesn't have repeating units. It has like kind of like components that you put together. But the point is, they are like a really big molecule that consists of, you know, multiple parts that you put together. Okay, so the individual parts, the individual components that you use to build the macromolecules, they are called monomers. Okay. And you can link the monomers together to form the polymers. Okay, so polymers are like examples of polymers would be macromolecules, but it's not restricted to macromolecules. Okay, you have polymers like all over your house. Okay? Plastic is an example of polymers, right? Your clothes, right? Sometimes they say it's a polyester, right? You probably heard of that, right? Your clothes is made of polyester. That's a polymer. Okay. So nylon, that's a polymer. Right? So in general, polymer just refers to very long chain of molecules that consist of repeating units. Okay. So in a biological system, the 4 things that we talked about, right? The carbohydrates, the proteins, and the nucleic acids, right? These are polymers. Lipids, not so much. Okay. So how do you link the polymers together, the monomers together to get the polymers? You can use a method called dehydration synthesis. Okay, or a condensation reaction. They mean the same thing. Okay, so condensation or dehydration means you remove a water molecule. So when you remove a water molecule, you will form a covalent bond that links the monomers together, and you end up having the polymers. Okay, when we eat the polymers, when you eat your rice, pasta, you're eating carbohydrates, your body will break down the carbohydrates. It will break the polymers back into the monomers. And to do that, you will use what's called hydrolysis. Hydro is water, lysis is to break. So you will break the chain using water molecules. Okay. So that is what it is. As always, if you have questions, if you don't know what I'm talking about, please interrupt me and ask. Okay. One of the things about online learning is I cannot see your faces, so I cannot read. Usually I can read student face and see whether they're confused or not. Now you have to tell me if you're confused. So let's start with carbohydrates. Okay. Elements contained in carbohydrates. So we have carbon, we have hydrogen, and we have oxygen. Those are the three elements that makes up carbohydrates. Okay. What about the building blocks? What are the building blocks for carbohydrates? Anybody? No? Anybody remember what the building blocks for carbohydrates are?
Glucose.
Very good. There is glucose. That's one building blocks. Anyone else? There are two more that we talked about in class. Start with an F. Anybody?
Fructose.
Fructose, that's correct. Okay. And one more, I'll do the last one, that is the galactose. Okay, so glucose, fructose, galactose. Okay, and it's probably worth mentioning that these are all isomers. Okay, isomers. Okay, meaning they have the same chemical formula, but different structure. Okay, so they are all C6H12O6, but then their configurations are different. Think of like a Lego, right? You buy a box of Lego to build a car or something. You can take it apart and build it into a boat, right? It's the same Lego pieces, same atoms, but different configuration. So that's what these are: glucose, fructose, and galactose. Now, what do we use carbohydrates for in a biological system in the body, right? Okay, so mostly for fuel. Okay, so fuel. It could be storage form of energy. Okay. So in humans, we store it as glycogen in the liver and muscle. In plants, they store it as starch. Not in humans, but in some organisms, carbohydrates can also use for structural support. Structural support. Okay, such as cellulose in plants. Okay, so fuel would be glucose. All right. We don't actually eat a spoon of glucose. Like, I mean, I don't think you can buy glucose off the shelf. We say glucose is sugar, but that's actually not entirely true. It's a type of sugar, but it's not the sugar that you will find in your kitchen. Anybody know what is the name of the sugar that is found in your kitchen? Like when you just buy a bag of sugar, what exactly is this stuff? Anybody know?
Sucrose.
Sucrose, that's right. That is sucrose, right? So sucrose is actually made up of glucose and fructose linked together, right? So when we put that into the body, we will use hydrolysis to break it down, and then you will burn the glucose for fuel. Okay, so that is that. Okay, using the diagram below, show how a bond is formed between two glucose molecules. So here is a glucose, here is a glucose. You are not responsible for memorizing the structure. I will never show you a picture of something like this and ask you, what is it? Okay, I will not do that. Okay, so how do we make a bond between the glucose here and the glucose here. Okay, so if you count all the carbons and all the oxygen, hydrogen, you would have 6 carbon, 6 oxygen, but 12 hydrogen. So if I put 2 of them next to each other, what's going to happen? Well, what's going to happen is there's going to be an enzyme. An enzyme is something that will help speed up or catalyze a reaction in the body. So if you put 2 glucose next to each other and you have the right enzyme, the enzyme will take a hydrogen from one of the glucose and it will take a OH from the other glucose, and then it will rip them out. And as a result, when you take the H and the OH away, you will get H2O. And then the carbon here will now form a bond with this carbon. Okay, so this new bond is going to be what's holding the 2 monomers together. Does that make sense? I sure hope it does. Let's do a poll question. Okay. Now, never mind what the hexagon and pentagon means. It just means there are 6 carbons here and 5 carbons here. But if I have 2 monomers showing like this, based on what we just discussed for dehydration synthesis, which of the following would be the correct result? Okay. Go ahead. Okay, waiting for 4 more responses. Okay. So most of you got it right? So you basically will take the OH from one of them, take the H from the other, and then you will free to connect them together. Okay, and the answer here is B. Very good. Okay, back to this thing here. Okay. So I guess here, like it was a little bit confusing that I put the C and the C. Actually, it is going to be like, you know, I actually took the H from the wrong guy here. I should take the H from here, like this, and then the O will join with the C here. Okay, so you get like a COC. Okay, but the key point here is, like, without going into too much of a chemistry, the key point is we will remove a water molecule and you will form a new bond. Okay. Next, complete the reaction. Okay, so if you take 2 glucose together, you will get maltose. Okay? If you look at the back of a candy bar, any candy bar, you will almost always see maltose. As we mentioned earlier, sucrose is a combination of glucose plus fructose, and lactose is glucose plus galactose. Okay. So these things over here, these are what we call disaccharide. Right? Saccharide is sugar, so these are like double sugars because it contains 2 monomers. And these things here, they are called monosaccharide. Monosaccharide. Okay, so if you take the monosaccharides and you put them together, you get the disaccharides. And then if you keep on adding more and more sugar on it, then you get a polysaccharide. Okay. Moving on. So here are some polysaccharide: starch, glycogen, cellulose, and chitin. Okay, so where do you find starch? Okay, starch would be found in plants, and you will find them, for example, in the roots. Right? So potato is part of the roots of a plant, right? They store all the starch there. Supposedly is for the plant to use later on, but of course we harvest it and we eat it. So the main purpose of starch is storage form of energy in plants. Okay. Glycogen. We will find it in muscle. Well, we say animals. And we will find it in muscle and liver. And that is the storage form of energy in animals. So not just humans, like, dogs or whatever as well. Pigs. Cellulose. What are cellulose? Cellulose are fibers. Okay, plant fibers. Plant fibers. Okay. So what do? They provide structural support. Right? They form cell wall. So we haven't learned about cell yet, but cell are these little things that makes up all living organisms, like trillion cells. And for plant cells, they would have like a cell wall outside. Okay. So the cell wall is made up of cellulose. Okay. Chitin. Okay, chitin is a special type of polysaccharides. It has a little bit of nitrogen in it as well. So carbohydrate is just CH and O, right? But then for chitin, there's also nitrogen in it. And they make up the exoskeleton of insects, bugs, right? Or crustacean. Okay, like lobster, crabs, sea-shelled animals. So like, you might have a parent or like a older relative that takes glucosamine. Okay, they can buy these little, what's the brand? Like Weber or something like that. Those nutrients jar, right? You can take calcium supplements, whatever. And one of the supplements that older people sometimes take is glucosamine. Okay, and that's supposed to be good for your bones. So the glucosamine is essentially extracted from these things. Okay, and that's one of the key structures of chitin. So what do? Well, obviously, because it's exoskeleton, it provides structural support and protection. Okay, that is that. All right, moving on to lipids. Any questions for carbohydrates before we move on to lipids? Anybody at all? No? Okay, let's do a quick poll here and see what happens. Go ahead. Oh, I'm sorry. I launched the wrong poll. Let me try that one more time. Okay, you're supposed to select all the false statement. So there are more than one possible answer. Okay. So let's see. First one, polysaccharides are important both for energy storage and to provide structure for cell. So that is correct. Okay, we're looking for the false statement. Storage because like starch, right? And glycogen, and structure like cellulose and chitin. Glycogen is important for both energy storage and to provide structures for cell. That is not true. Okay, that is not true because it doesn't provide structural support. Cellulose is only found in animal. That is not true. In fact, cellulose is only found in plants. Glucose, fructose, and galactose are examples of monosaccharides. That is correct. Starch is the storage polysaccharide made by plant cells. That is also correct. Okay, starch is the storage form of energy of plants, right? So the answers for this question would be both B and C because those are the false statements. Okay, what is this thing? Okay, let's move on, discard, and go back to here. Okay, next we have lipids. Lipids, what's going on with lipids? Now, lipids is a little bit different because, and I think I should write this down for you, okay? Lipids is a little bit different from the other three types of macromolecules because lipids lipids are not made up of repeating monomers like the other three types of macromolecule. Okay. Instead, it has components. Okay, it has components that you put together. It's like building a computer. So we have different types of lipids, and there are three types that is being shown here. The first one is fat. Okay, fat. Okay, there are two types of fat. Okay. And fat has another name, right? I think I talked about it in the lecture. I don't remember. Triglyceride. Okay, so there are two types of fat. There are saturated fat and unsaturated fat. Okay, and we will talk about the difference and similarities later on. But this one here, this picture shows a saturated fat. Okay, but nevertheless, they are fat. Okay, saturated, or whether it's saturated or unsaturated, the components are the same. So how do we make a fat molecule? Well, you're gonna need to have two things. You're gonna need to have one glycerol plus 3 fatty acids. Okay, so just a little extension for anybody who cares. Glycerol looks like this. Okay, it's called the ho molecule. Okay, so over here, it's more like O instead of ho, but there you have it. That's the glycerol. And then you have the fatty acid. Okay, and again, I don't expect you to know how to draw these. Okay, so you have a bunch of C's here, and the C's would be connected to H. Okay, but what I want to reiterate is if you want to join them together, you're going to need to have an enzyme. And then what's going to happen is the enzyme will remove OH from one, remove H from the other, and you will release a water molecule, and then you will be able to form a new bond, right? New bond like this. So to form a fat molecule, you will need to have one glycerol, and then you will have to have 3 fatty acids. So here's one, you have another one and another one. So you will release 3 water molecules. Okay, so that's just to reiterate some of the things that I've said. What is the main function of fat? Well, fat is important for storage form of energy, right? More like long term compared to glycogen. Glycogen is short term. Okay, like if you didn't eat breakfast this morning, right? Like you just got up and you're like, oh, I gotta go to a lecture. You did not eat breakfast. Right now, you're burning your glycogen reserve. Okay, those are short term storage. Right? If you don't eat for a couple days, okay, for whatever reason, okay, then you will start to burn off your fat reserve. Okay, in fact, as little as like 16 hours into fasting, you will start to burn off some of your lipid reserve. Okay, but fat is also good for insulation and protection. You will often find lipids around like organs. They will keep them warm in the winter, insulate, and it will protect them if you fall on the ground. They're kind of like bubble wraps, right? Kind of cushions the shock a little bit. So that is the fat. Okay. Next, we have what we call like a phospholipid. Phospholipid. So the phospholipid has 3 parts to it. Okay. There is a polar head here, a polar head. Okay. Which is made up of phosphate. And then we have the hydrophobic tails. Hydrophobic tails. Okay, so this part is water-loving, hydrophilic, and then this part is hydrophobic. Right? So it makes the molecule have 2 distinct properties on different ends, and that's going to be important when we talk about things like cell membranes. Okay, so what do you need? Well, we're going to need 1 glycerol, just like the lipid. It's right here, the molecule. We're going to need 2 fatty acids this time instead of 3. 2 fatty acids, and then a third slot that used to be a fatty acid. Well, now we're going to put a phosphate group on it instead. Phosphate. So PO4 -3, right? You might remember learning that in chemistry. What do we need the phospholipids for? Well, one function is to form cell membrane. Right? Cell membrane is what surrounds your cells. It separates the living environment on the inside of the cell from the non-living environment on the outside. So that's the function. Next here, we have a bunch of rings. Each of the corner of the ring represent like a carbon. Okay, usually people don't draw them. So this is an example of a steroid. Okay, steroid. So our body makes natural steroids, and steroids basically have these 4 distinct carbon rings. Okay, so these are here carbon rings, and then we have these things that we add on to them. They are called functional groups. Functional groups. Okay, like think of the rings as like your phone, and then the functional groups would be like the accessories that you put on them. Okay, if you put on different accessories, you're gonna have like different function. Okay, it's gonna change how the molecule is going to behave. Okay, so what do we need? Well, we need 4 carbon rings. Carbon rings, right? 1, 2, 3, 4. That will be the core structure plus various functional group. Functional groups. Okay, so example of a simple functional group is an OH. If you add OH to a molecule, right, it gives it alcohol property. OH is actually called alcohol, right? So function of steroids, right? We use it as hormones in the body. Any examples of hormones? Any do you guys know any sex hormones, male, female hormones in the body? Anyone?
Estrogen.
That's right. So estrogen is a steroid hormone. Okay. Testosterone, right, for male, that is another steroid hormone. And you make them from cholesterol. Okay, cholesterol. Cholesterol is the starting material for estrogen and testosterone. We can use it for water repelling surface. Water repelling surface. So like if you think about like beeswax, that is a steroid. All right. Any question about the 3 types of lipids? You guys still with me? Are you still there? It's hard to tell. Moving on.
Can we go over
Yeah.
the second lipid with the polar head?
This one?
Yes.
Okay, so what's the question again? Come again, please.
I don't understand what differentiates it from the first one. The first one has
Okay, yeah.
cholesterol.
Right. So the
And-
first one has 3 tails, right? 3 fatty acids. Right. So this is going to make the entire molecule hydrophobic. Right? That's why fat will not dissolve in water. You put butter in water, it will not dissolve. Butter is an example of fat. Phospholipids, on the other hand, because instead of having a third fatty acid tail, that third slot is used to connect to a phosphate group. So phosphate, the chemical formula for phosphate is PO4 with a charge of -3. If I zoom in here, right, you can see there is the negative, right? There's 1 negative here, and then you don't really see the other negative for reasons that we're not going to go into. But the point is with the negative on the phosphate group, this part of the molecule is going to be charged. Okay, and if it's charged, then it's going to be polar because the water molecule will now be able to bind to it, right? Specifically, the hydrogen part of the water molecule will bind to it. So by switching out a tail with a head, we make a phospholipid. Okay, that allows it to interact well with water. This here does not interact well with water. So that's the key difference. Hope that answers your question. Feel free to have follow-up questions You can type it in chat. Moving on. Compare saturated and unsaturated fats by completing the table below. Okay, so saturated fat, right? What is saturated fat? We would have 1 glycerol plus 3 fatty acids, just like before. Unsaturated fat is exactly the same. Exactly the same. You would have 1 glycerol and 3 fatty acids. And fatty acids is that long zigzag thing, right? And again, what is the zigzag? The zigzag is just a bunch of carbons, right? Okay. And when you learn organic chemistry and chemistry in your chemistry class, you will see, you will learn how to draw these zigzag things. Okay. So types of fatty acids. Okay? So the types of fatty acids here would be what we call saturated fatty acid. What does that mean? Well, it means there will be no double bond in it. No double bond. And then the carbons are bound to carbons are bound to maximum number of hydrogen. Okay. And again, I don't know how far you guys are in chemistry, but carbon can only have 4 bonds, right? Carbon can only have 4 bonds. Did you guys learn that yet in chemistry? Just say a yes or no. Let me know. Do you guys know carbon can form 4 bonds? Yeah, okay. So if you have 2 carbons next to each other, right? Okay, like if you think about this carbon right here, right? It will take up 1 bond with 1 carbon and then another bond with another carbon. So it has 2 more slots. Okay, so both of those slots will be hydrogen. So now it's saturated. There's no more spot for more things to bind to it, right? So it's bound to maximum number of hydrogen. We call that saturated fat. Okay. On the other hand, the unsaturated fat, right? The unsaturated fat, you would have saturated. You would have a mix of saturated plus unsaturated fatty acid. Okay, so up to 3 unsaturated fatty acids. Sometimes it's 1 unsaturated. Remember, it has 3 tails, right? So sometimes you have 1 saturated and then 2 unsaturated, or 2 saturated, 1 unsaturated, or you can have all 3 of them being unsaturated. Okay, so here you're gonna have some double bonds. Okay, some double bonds, and then carbons are not bonded to max number of hydrogen. There's still room for more hydrogen. Okay, so there is something called hydrogenation. Right? That's how they make like margarine and stuff. They add hydrogen to unsaturated fat, and it converts them into like a semi-solid state that you can spread them. Okay, so that's called hydrogenation. So what's going on with this? Well, if it's saturated fat, because it doesn't have double bonds in the fatty acids, the tails are straight. So you can pack them really closely like that. Okay, there's not much space between the molecules. But if it has a double bond, right, it will cause the tails to have a kink. Okay, because of the kink, it will cause the molecules to be further apart. And so if it's really, really packed, then at room temperature, this stuff is going to be solid. Okay, so like butter. Butter is solid at room temperature. Okay. Unsaturated fat, on the other hand, it's going to be liquid because they don't pack really well together. Okay, so canola oil, olive oil, all those things are liquid at room temperature. Natural source, most saturated fat, most are derived from animal. Okay. There are exceptions like coconut fats. Coconut fat is actually saturated, even though it's a plant base. Okay, but most of the saturated fats come from animals. Okay, like lard, bacon grease, all those things are saturated fat. Unsaturated fat would be most are derived. Derived means come from, right? Derived from plants. Okay, so canola. Excuse me, canola oil, right? Olive oil, grapeseed oil, whatever oil you use, peanut oil. Okay. Questions before we move on? All right. Next, we have proteins. Okay, so label the different parts of the amino acid below and show how peptide bonds is formed. Okay, so carbohydrates, the building block for carbohydrates are like glucose, fructose, galactose, right? For proteins, the building blocks would be the amino acids. So there are 20 different types of amino acids, but they all kind of share the general structure like this. Okay, so there is a central carbon in the middle. They all have that. Okay, so this is the central carbon. All right. There's going to be hydrogen connected all the time. Over here, you have the NH3 is called the amino group. Amino group. And again, you will learn more about this in organic chemistry. I think that's next semester. And over here, this is called the carboxylic acid. So because it has amino a group on one side and an acid on the other side, this is called an amino acid. Amino acid. Okay. And then there is the R right here. The R. What is the R? R is called the side chain. Okay. And then there are like 20 different side chains because there are 20 different amino acids. And depending on what the side chain is, it will give it different properties. Okay, so sometimes the side chains are polar, sometimes it's going to have a positive charge, sometimes it's going to have a negative charge. So if you put two amino acids close to each other, one having positive charge, the other having negative charge, they will be attracted to each other. Okay, so that's one example of how the side chains can interact together. So remember, these are the monomers of proteins. So if you put one amino acid next to the other, and then you bring in an enzyme to hook them up together, then what happens is the enzyme will, just like we said in the other cases, it will take an OH from one group, from one amino acid, from the carboxylic acid of one amino acid, and then it will take a hydrogen, and then it will become H2O. Okay, so what would that look like afterwards? Well, it's going to look like this: NH and H, carbon, hydrogen, and this would be the side chain, and then C double bond O, and then this C will now connect directly with N. Okay, so this is another R, and then you're going to C double bond O, OH. Okay, like this. So this here is the new bond. We call it a peptide bond. Okay, and then we could keep on adding more. We can add another amino acid and another, so on and so forth. So it forms like a long chain. All right. So you know what? I'm just gonna add another other one to show you. Okay, so you would remove an OH and then you would connect it with another H. Right. and then you would have an other R. So this would be side chain 1, side chain 2, side chain 3. And I think what you can see here, I hope what you can see here, is that this is basically repeating over and over again, right? Okay, so this yellow part here, this is called a peptide backbone. Okay, and it's just going to be the same thing over and over again for the entire protein. What differs is the side chain. Okay, so these are the different side chains, right? Side chains. And each time you create a peptide bond, okay, let me use a different color here. Each time you create a peptide bond, but there's one peptide bond here, another peptide bond, right? Peptide bond, you will have to remove a water molecule. So to connect 3 amino acids together, I would be removing 2 water molecules. Okay. All right. If you need to take a picture or a screenshot or whatever, you can do that. Moving on here. Complete the table about proteins. Okay, so what kind of elements do we have in proteins? Well, we have carbon. We have hydrogen, oxygen, nitrogen. Okay, and then we actually have traces of sulfur. Okay, so of the 20 amino acids, one of them has sulfur in it. Okay, something called cysteine, C-Y-S-T-I-N-E. What's the building block for proteins? Amino acid. And like I said, there are 20 different types. Okay, so most of it your body can make. There are 8 of them that you cannot make, and you must, obtain it through your diet. Right, so I mentioned that in the lecture recording. What are the biological functions of proteins? Well, there are so many different functions. For example, there is enzyme. Okay, so someone just messaged me and said, yeah, if you need to go for whatever reason, you can go. This will be available as a recording, okay? And I'll post an announcement afterwards, okay? So you will be able to find it, okay? Biological function, enzyme, right? Enzyme help catalyze reactions, right? It speeds up chemical reactions in the body. We also need protein for structures. Okay, so your muscle is actually made up of proteins. Your hair is made of proteins. Your fingernails, they are made of proteins. Right? Horns, we don't have horns, but then like if you think about animals, those horns are made up of proteins as well. We can use it for hormones. Okay, so earlier we talked about estrogen and testosterone. Those are hormones, but those are made up of lipids. There are also hormones that are made up of proteins. Okay, so one example is insulin. You guys know insulin? Okay? People need insulin to, absorb the sugar from the blood after a meal. If you cannot make insulin, or if you don't respond to insulin, then you might have diabetes. Okay, and we can also use protein as fuels. Okay, so this is like last resort. Like I'm stranded on an island for 7 days. Okay, I have no food at all. So at the beginning, you will burn your glycogen reserve, and then you will start to burn your fat, and then you will also start to burn your proteins later on. Okay, so you waste away your muscle, lose weight, because your body is breaking down the proteins and using the amino acid as food. Okay, so that's proteins. Next one. Now, when it comes to protein, there are 4 different levels, right? That helps contribute to the overall shape of the protein. The key thing here is that the proteins, in order for them to function properly, they must be of a particular shape. Okay, it is very sensitive. If you change the shape of the protein, they will either not work at all, or they will work in a like a different way. It will work in a inappropriate way that would cause some kind of problems. Okay, so what happens is it's organized into 4 levels of structure. So the primary structure of a protein, when someone say the primary structure of a protein, they are referring to the sequence of amino acids. So like I said, right, there are 20 amino acids. And when you build a protein, the instruction to build a protein is actually embedded within your DNA. And we'll learn more about that in later units. But your DNA basically tells your cells what amino acids should link together. So, you know, if I take amino acid, like if I take a purple amino acid and then I link it to like what color is that? Pink, and then green, and then maybe another green, and then blue, red, whatever, right? So on and so forth. This sequence of amino acid, the order at which I link them together, that is called the primary sequence. Okay, so obviously amino acids don't have colors. They would have names, right? So, you know, this would be maybe like alanine, like phenylalanine, whatever, glutamic acid, and so on and so forth. So if you read the identity of each of the amino acids, the order at which you read it in, that is called a primary sequence. Okay, so the primary sequence of amino acid is determined by our genetic code. Okay, so if you took biology in high school, you might remember, you know, how that works. And if you don't remember, or if you haven't taken biology before, that's okay. We will learn about it in, I don't know, lecture 6 or 7, something like that. We'll see. So what holds the amino acid together? The peptide bonds, right? The peptide bonds. Remember up here, right? What is holding this amino acid together with this one? Well, it is the peptide bond in between. And you form the peptide bond by removing a water molecule. Okay, so you can see a theme here, right? By removing water molecules, we are able to join the monomers together. Next, we have secondary structure. What is secondary structure? So secondary structure is either we have these alpha helix. Okay, this symbol means alpha. Okay, and then this is beta. And beta pleated sheets. Okay. So what that means is here we have the primary sequence, and then the primary sequence, depending on the type of amino acid, some section will coil up to form these beta sheets, and then others, form these alpha helix, and then other parts would form these beta pleated sheets. Okay. So here, alpha helix would be like these parts, and the beta pleated sheets will be like these parts. Okay. And what's holding these sheets together is actually a hydrogen bond. And we talked about hydrogen bonds earlier with the water molecule, right? Okay, the slightly negative part will be attracted to the slightly positive part. Okay, but it doesn't have to be water molecules. It could be other molecules as well. In this case, it would be the hydrogen bonds of the peptide backbone. I showed you what the peptide backbone was earlier. So this, for the most part, is still relatively 2D. It's like flat on a piece of paper. Then it will continue to fold. Okay, so maybe this will fold over like out of the page, and then now you're gonna get this. I can't really draw this too well, but then it's like a big mess, right? Okay, and you have like a 3-dimensional shape. Okay, 3D shape now. So that 3D shape is going to be the tertiary structure. Okay, tertiary structure. So that is the overall 3-dimensional shape of the protein. And I cannot stress this enough. The 3D shape is what dictates the function of your protein. You don't want to mess up the shape. Okay? If there is a mutation in your DNA, in your genetic code, that causes the shape to be different, then you're going to have a problem. So in the lecture, right, I mentioned something about sickle cell anemia. That's just one amino acid change in the hemoglobin sequence, and then that causes your red cells to become moon shape, and then you will have anemia, where you cannot deliver oxygen easily. Okay, another example that I showed you, I think, is a mad cow disease, right? Okay, so we have a normal version of the mad cow protein, it's called the PRP protein, prion proteins, and that one has a lot of helix, but the mad cow prion is the same protein but misfolded. It has a lot of beta plated sheets in it, and then the beta one, the misfolded one, can cause the normal one to misfold as well. It's kind of like a bad influence, and over time you will have all these misfolded proteins, which will cause holes to form in your brain, and that of course is not a good thing. Okay, so having the right shape, the right tertiary structure, is vital for the normal function of the protein. So what holds the 3D shapes together? It is the interaction between the side chains. Okay, so the side chain is the R, right? Okay, depending on which amino acids we have, that's going to dictate the overall 3D shape of the protein. Any question before we move on? You guys are awfully quiet.
Sorry, I wanted to go back to question number 14.
Of course, go ahead.
Just between the two, when we're labeling it, between the two of them, did you want that to say dehydration synthesis?
Oh, this blank?
Yeah.
Oh yeah, that I just, it was supposed to be H2O on it, but yeah,
Oh.
you're right. It's dehydration synthesis, exactly.
Okay.
Yeah.
That's it.
There we go. Okay. Is there any way you can post the notes? You guys want this stuff to be posted? Yeah, sure, I can do that. I mean, it's just chicken scratch, right? It's but if you want it, sure, I'll post it. Okay. Yeah, most of the stuff is directly from the lecture, anyways. But sure, I can do that. Quaternary structure. Quaternary structure. What is quaternary structure? So if you have several proteins with 3D structures and they come together, right? So this might be like, we call like, you know, peptide 1, and then you have another peptide 1, here peptide 2, and then it comes together, right? What peptides you bring together to build the overall thing, right? That is the quaternary structure. So when 2 or more polypeptide is just like one protein, right? Join together to form what we call a protein complex. Okay, so not all proteins are going to have a quaternary structures, only some of it. Okay, so an example of something that has multiple polypeptides, right? It's a hemoglobin. Okay, hemoglobin helps you carry oxygen in the body. Okay, so it has 4 subunits, 4 polypeptides in, right? 2 alpha and 2 beta. Okay, so what holds it together? It is interactions between each polypeptide. Okay, so maybe there is some bonding here, just holding it together. Okay. All right, let's quickly do another poll. See if you are with me? Go ahead. Okay, please give it a try. So this one's kind of all over the place. We have people answering for many different things, but let's take a look. Pancreatic cells make insulin, which is a type of protein. That's the keyword. Okay, so these cells use something in order to synthesize insulin by the process of something. So proteins, we just talked about what are proteins made up of. Well, proteins are made up of amino acids, right? And to connect amino acids, we do dehydration synthesis. So the answer is E. Okay. Some people choose B, right? Fatty acids, glycerol. That's for lipid, right? Okay, not for proteins. I hope that makes sense. Okay, let's see. Last part here now. Nucleic acids. Complete the table about nucleic acids. So what is a nucleic acid made up of? Well, we have carbon, we have hydrogen, nitrogen, oxygen, as well as phosphorus. Okay. The building block of nucleic acid is called nucleotides. Nucleotides. And a biological function of nucleic acids, well, it encodes for genetic information. Okay. And it has the instructions for making proteins. Okay. Next, identify the phosphate group, sugar group, and nitrogen base of a nucleotide. A nucleotide is going to have three distinct parts. Okay? And you can see in this picture, the three distinct parts are the sugar here, sugar. Okay. It is going to be a ribose sugar for RNA. It's going to be a deoxyribose for DNA. Okay. It will have a phosphate group. Okay, which makes DNA negative. Like the PO4 -3, right? And then it's going to have this nitrogen base. Nitrogen base. Okay, so together, this makes a nucleotide. Okay? Nucleotide, which is the basic building blocks for nucleic acids. Now we can classify the nucleotides into purine and pyrimidine. Okay. So over here, these three things, right? Again, I won't ask you for the structures, right? But the C cytosine, thymine, and uracil, they are the pyrimidine. And then the other ones over here, the adenine and the guanine, that's how you say them. These are the purines. I could not memorize them when I was learning this. Like for some reason, I just couldn't remember which one is which. And then someone told me, you can remember it like this. Pure as gold. If you remember this phrase, then you can remember it. Pure as gold. So purines are adenine and guanine. Okay, everything else, pyrimidine. So you can see purines have 2 rings in their structures. Okay, 2 rings. Whereas pyrimidine, they only have 1 ring. So listen for a second. If you think about the DNA, you should know by now, DNA is like double helix, right? So it's going to have like 2 strands on each side. And then you're going to have the base in the middle, right? So like A, T, for example, G, and C, like this, right? And then so on and so forth. In order for us to maintain a uniform width along the DNA, you must pair a purine with a pyrimidine. Okay, because purine is going to have 2 rings. So like, 1 ring, 2 rings of this wide. And then a pyrimidine is going to have 1 ring. So if you put them together, the space is like 3 rings equivalent of width, right? So you will always have a purine with a pyrimidine to ensure you maintain that consistency. You cannot, for example, pair a purine with another purine, because then that would be 4 rings across. It will be too wide, right? And you cannot pair a pyrimidine with a pyrimidine, because then you now only have 2 rings and it will be too narrow. So throughout the entire length, you want to maintain a distance of 3 rings, okay? So 1 purine will always pair with 1 pyrimidine. And in fact, I'm sure you know by now, A pairs with T and G pairs with C, okay? And then the uracil, there's not much pairing going on because that's only found in RNA. Are we okay? Do you understand what I just said? I hope so. Oh, good. Some of you are giving the thumbs up. That is actually a great feature. It's just a quick way for me to see if you're with me or not. So if you could use that, that would be wonderful. If you click on the ribbon, there is a reaction button, right? And then you can choose a reaction. Okay, DNA versus RNA. Nucleotide. What is the nucleotide made up of? Well, we are going to have a phosphate. And for DNA, which stands for deoxyribose nucleic acid, so we're going to have a deoxyribose. It is a sugar missing an oxygen plus a nitrogen base. Whereas the RNA, we are going to have a phosphate, and this time instead of the deoxyribose, we have the ribose sugar plus nitrogen base. I don't know if you noticed, but anything that ends with OSE is like a sugar, right? Think about glucose, fructose, galactose, they all end with OSE. And now we have a ribose, okay? What kind of nitrogen base do you have for DNA? We have the A adenine, thymine, cytosine, and guanine. But for the RNA, instead of having the thymine, we will have the uracil instead. Everything else is the same, so no thymine. Strand structure, this is a double helix, which means it is, you know, basically the generic thing that you see on TV, okay? And then you will have the base here in between. And for the RNA, it is single strand, which looks like this. Okay, now it's possible for the single strand to fold up on itself. Okay, so like, you know, some of the RNA, it's gonna fold up like this and form like a clover leaf shape, right? This is something called tRNA, right? We will talk about that later. Okay, but it will be able to fold up onto itself. So this part would interact with each other, right, to stabilize the structure. Okay, so more on that later. And that is it, okay, for the nucleic acid. Now here are some long answer questions. I will leave it on here, and I want you guys to let me know which one you want me to take up. I mean, we only have, what is it, like 25 minutes. It's not possible to go through all of them. So, you know, which one would you like me to discuss in more detail? Please let me know. Give you a moment to read through them. No requests? Okay, number 1.
For number 5, the differences between DNA and RNA. I know we would use like the table that we just completed in number 20, but do you want us to like explain anything in terms of function?
That's a good point. Did I talk about any functions in the recording? I honestly don't remember. But yeah, let's. Sorry, did I?
I don't think so. No.
No. Okay, that comes later. Okay, so
Okay.
probably is okay. Yeah. All right. Okay, so just number 1. Well, number 1 is quite easy. We can just go back to the chart, right, that we did at the beginning, right? Okay, the various polysaccharides and state or biological function. Where was that? There is a chart right here, right, number 11. Okay, so these are all the polysaccharides, and tells you where to find it and their main biological function. Okay, so that's just, you know, sometimes writing it out in a chart helps you organize your thoughts, but if you write it out in a paragraph, that will help you, you know, consolidate. You know, like this study guidebook was created for like when we were doing face-to-face. So these are some of the questions that you might get on a test. Okay, but obviously online, I'm not going to ask you to have written response, because what most people end up doing is just copying pasting from the notes, right? So there's no point of doing that. So that's why you see these questions kind of repetitive. But you know what? You know, when you go to next semester, second semester, I teach the anatomy. I told you, maybe, hopefully, fingers crossed, we will finally meet face to face, and you know, these questions will be more useful by then. All right, I think I will talk about number 3. Nobody asked for number 3, but it's actually quite important. Okay, enzymes and their substrates have a key and log relationship. Explain what this means. Okay, so I'm going to do that. Okay, I'm going to do that right here, number 3. Okay, so first of all, like, what is an enzyme, right? So enzyme, they are biological catalysts. Okay. A catalyst is something that speeds up chemical reactions. They speed up chemical reaction in the body. Take sucrose, for example. Sucrose. Okay, so sucrose, as you know, is made up of glucose connected to our fructose. Okay, so I'll use square to represent glucose and triangle to represent fructose. If you leave your sucrose in the kitchen, it is not going to break down into glucose and fructose. Okay, because there's no enzyme catalyzing that reaction. It's just going to stay as sucrose for until the end of times, pretty much. But when you eat the sucrose, put in your body, then you will have enzymes that will help you break it down. So an example of an enzyme that breaks down sucrose would be sucrase. So anything that ends with ASE would be an enzyme. So to break down lactose, you would need lactates, right? And if you don't have that, you might be lactose intolerant. So over here, sucrose, you're going to take sucrase, the enzyme, and it's going to break it down into glucose. Why is my color not changing? Glucose plus fructose. Okay. So the enzyme sucrase is going to look like this. First of all, the enzyme is going to be made up of proteins. Okay, so this is sucrase, and it's made out of protein. So it's going to have a three-dimensional shape. Okay, and what's important is that there is a particular part in the protein that we call the active site. Okay, active site is where the substrate is going to fit in. What is a substrate? So this sucrose rather, it's going to go into the active site. So this is the substrate. The substrate is the thing that the enzyme is going to be working on. Okay, it's the material, starting material that the enzyme is going to cause a reaction to occur. So what's going to happen is the sucrase will fit into the box, into this active site, and then the enzyme will break the bond, and then it will release the glucose and the fructose. So there is a key and log relationship between the active site of the enzyme and the substrate, which means if I have, say, lactose. If I have lactose, okay, lactose. So lactose is glucose plus galactose, right? So instead of triangle, I'm going to use like circle. So this will not fit in sucrase, okay? Because it's the wrong shape, right? Just like if you have a different key, it won't open the lock, right? That's what it means by key and log relationship. Okay, so in order to break down the lactose, I would need to have another enzyme, the lactase, which has a different shape in the active site, and then they would have a relationship and be able to break it down. Okay, so that's what key and log relationship means. Also, if you have a mutation, right? If your genetic code changes and your protein is misfolded, right? That's like bending the key. You will no longer be able to, you will no longer be able to open the lock if you bend the key. In the same way, if you change the shape of the active site, either because of mutation or because, if there is temperature fluctuation in your body, that's why like a really high fever for a long time is dangerous, because that high temperature can change the shape of your proteins, right? And then they will no longer work. Okay, so that's what it means. Any follow-up questions? That makes sense, hopefully. Okay, let's quickly do the fill in the blanks and then I will let you go. I mean, you're free to go anytime anyways. I'm not holding you hostage, but you know what I mean. Okay, atoms can combine in fixed proportion to form molecules. Molecule is the smallest unit of an compound. Oh, sorry, I forgot. There is actually one more poll that I forgot to do. Let's do that first before we finish with the fill in the blanks. Go ahead, try that. A lot of you are actually not doing the polls. Is it because you guys cannot see the poll? Because sometimes that happens. Or you guys just like reading the questions. Can you let me know what it is so I know what the problem is and I can fix it? Are people not seeing the polls? Okay, all of a sudden there are more people answering. Okay, there we go. So what's the right answer? Yeah, right answer is the last one, but let's take a look at the other things here. Okay, adenene's bonding to thymine is stronger than guanine to cytosine. That is not true. It's actually the other way around. I didn't mention it in the tutorial, but I'm pretty sure I did in the recordings. But if I didn't, A and T has 2 hydrogen bonds between them. 2 bonds, 2 hydrogen bonds, holding them together. Whereas G and C would have 3 hydrogen bonds holding them together. And this is going to be important when we talk about transcription and translation later on. Cytosine is a purine. Remember, pure as gold. So A and G only for purine, so that's not true. Purine binds readily to deoxyribose, but not ribose. That is just wrong on so many levels. I don't even know how to correct it. This is nonsense. Okay. Both adenine and thymines are purines. Same thing, pure as gold. Nope. And then double ring. We know that they're double ring. Okay. So just generally speaking, you like on the quiz, you should take no more than a minute and a half for multiple choice question, and no more than 2 minutes and a half for the multi-select and application questions. Okay, so that's like the formula we use as teachers to determine how much time we give you on a test. Okay, plus a little bit of buffers. So if you find yourself, really stuck on a question, maybe come back to it. Okay, because you can just skip the questions, right? Okay, like I know in high school, some teachers make it impossible to go back to like an earlier question. Like we're not gonna do that. We're not gonna be like, evil like that. Okay, so you can have a chance to go back later. So do try to, skip the ones that you don't know and then come back to it later. Okay, back to the fill in the blanks. Number 3, atoms is the smallest unit of an element. A metal and a non-metal can combine to form an ionic compound, whereas a molecular compounds, they contain 2 or more non-metal atoms. In a solution, the solute is the substance that is being dissolved. And the other one is a solvent. Substances that can be dissolved in water are called hydrophilic. Polymers can be broken into monomers by addition of water in hydrolysis. Two glucose molecules join in a condensation reaction or dehydration synthesis reaction to form maltose. Hydrolysis of sucrose will produce glucose and fructose. Storage form of energy in animal, glycogen. Liver and muscle. For plants, it is starch. Think potatoes. The fibers in plants are made up of cellulose. Main structural components of cell membrane, that is the phospholipids. Testosterones and estrogens are steroid hormones. Okay. There could be steroid hormones and there could be protein hormones, right? Double bonds in the fatty acid tail, that would be the unsaturated. That is the healthier form of fats. If you have to chuck down some fats, you would want to choose unsaturated fat. Something are proteins that catalyze chemical reactions in living organisms, that is enzyme. The general structures of amino acids contains an amino group, and acid group, right? Carboxylic acid, which is called acid for short. Conformation is the shape of a protein. When it unfolds, it is denatured. Denatured. The monomers of nucleic acids are nucleotide. Nucleotide. And the monomers of proteins are amino acids. 5 types of nucleotides, 20 types of amino acids. Fat molecules contains glycerol, the ho molecule, and 3 fatty acids. If the fatty acids have unsaturated fatty acids have double bonds in it, then it's an unsaturated fat. Otherwise, it's a saturated fat. Alpha helix and beta plated sheets are parts of secondary structure of proteins. In DNA-based pairing rule, thymine pairs with adenine, whereas cytosine pairs with guanine. Okay, so I'll post this afterwards as I promised. Just before you go, I like to reiterate for next week. Well, first of all, you have to do the quiz, right? And also you have to watch the recordings. Go to content, lecture 3. These are the lecture recordings, okay? Please watch them before you come to class. In addition, if you want to access previous tutorial recordings, the one that we do on Fridays, go to useful information. There is the tutorial recording, okay? So if you click on that, it will take you to the online page. So this is this was last week's, and then after class, I will post another one for this week's, okay? And also, for those of you who are new and don't know when things are happening, you can download the weekly schedule to see, okay? If you need extra help for the quiz, please feel free to email me. Otherwise, best of luck. Have a good weekend, and I'll see you next Friday. Okay, 8:30. Bye for now.
Bye.